लाइव कनेक्शन में जो हैं वो रिप्रेजेंट कर दे अपने आप को, कौन कौन है लाइव में जो जुड़ चुके हैं। अगर मेरी आवाज़ पहुंच रही है तो एक मैसेज कर दें, क्लियर हो जाए कि आवाज़ पहुंच रही है आप लोगों तक। अगर आवाज़ पहुंच रही है तो आप मैसेज कर दे ज़रा। तो आज बायो एनर्जेटिक्स का हम लोग नेक्स्ट टॉपिक पे आज हैं। हमने इन्फॉर्म भी कर दिया था कि आज हाई एनर्जी कंपाउंड्स पे हम लोग आज बातें करेंगे। और इसके पहले इन द ऑल प्रीवियस क्लासेस in all the three previous classes we will discuss on the different aspects of the bioenergetics, like what is the free energy, what is the enthalpy, what is the entropy, and what is the coupling reaction. And for the during the discussion on the coupling reaction, I was explained that the energy, released energy, is stored in the form of ATP, and which is responsible for the transformation and transportation of the energy in the living system. That means the most important high energy compound which we observe in the living system is the ATP. And we all know very well that the full form of the ATP is the adenosine triphosphate. And the adenosine triphosphate is a special carrier of free energy in the form of ATP. That is the special carrier molecules of the free energy. Then the energy released in the system is converted into the form of ATP, which is responsible for the transportation of the energy at the site of requirement, as well as it is responsible for the all vital metabolic energy where energy is required. Now, the ATP is played a central role in the transference of free energy from the exergonic to the endergonic processes in the cell. That means the exergonic represents the energy yielding, to the endergonic represents the energy required. That means the ATP is responsible for the formation of a bridge between the exergonic and endergonic reactions occurred in the living system. Then the you all studied in the intermediate level, at the intermediate level or at the postgraduate level, that the ATP is formed by 1 molecules of adenine, 1 molecules of ribose sugar, and 3 molecules of phosphoric acid. That is, adenine is associated with the ribose sugar at the 5' end by the 9th nitrogen and 5th carbon. And here it represents actually the glycosidic bond. Second, the first carbon is associated with the phosphoric acid, then 1 phosphoric acid is sequentially associated with 2 further inorganic phosphoric acids, and in this way it is responsible for the formation of one molecules of ATP. Then what is the difference here? ATP has two high energy bond and one low energy bond. Then what is the point of differentiation of the high energy and low energy bond? And in this condition, it is uh, said that the high energy bond generally, because in the ATP two high energy bond is present, and mm, commonly it is considered that the bond has more than 21 kilojoule per mole, more than 21 kilojoule per mole bond free energy nature, or contains the energy more than the 21 kilojoule per mole, is considered as the high energy bond. In some books, the 25 kilojoule is mentioned. So it is better to say that if the uh, energy is more than, if any bond has energy more than 21 kilojoule per mole, is represented the high energy bond. And here we are discussing at first the high energy compound is the ATP. So, in order to maintain a living process, all organisms must get free energy from their environment, because for the work in the living system we are completing numerous work, or from necessary for the activities of the living system or the living property. So in order to maintain living process, all organisms must get free energy from their environment. The autotrophs, there are two types of organisms or the living components in our environment, then autotrophs and heterotrophs. So the autotrophs get supply of free energy by coupling their metabolism so to some simple processes in their surroundings. And it is, we know that the autotrophs commonly obtain energy from the from the photonic form, in the photonic form, whereas the some chemoautotroph microorganisms obtain the energy from the biochemical molecule, or that is the bond energy. So mostly the autotrophs are responsible for the receiving of energy from the, in the photonic form, and it is the simple form for the conversion. And second is the heterotrophs, and the heterotrophs get free energy by coupling their metabolism to the breakdown of complex organic molecules. We all studied that the ATP is formed by the catabolic reactions primarily of the carbohydrate, but sometimes we obtain the energy by the catabolic activities of the lipids, and at last, and rarely from the proteins, in the condition of the more starved conditions. So, the in the autotrophs, the free energy is obtained by the catabolic activities of the complex organic molecules. Here, the ATP, in the ATP, the energy, high energy bond is present in the form of phosphate bond. And when we move today we will discuss that some other high energy bonds are also present in the living system, and phospho high energy phosphate bond. And the importance of a phosphate in the intermediating metabolism was realized by Fritz is the name of scientist, is, is realized by the Fritz in 1941 and introduced the concept of high energy phosphate and high energy phosphate bond. So it was Fritz who explained the concept of the high energy phosphate and high energy phosphate bond. Here in the ATP, the release and association during the normal metabolic reactions is associated by the magnesium ion. It is not released directly, but we will discuss today that the phosphate bond and the high energy nature is responsible for the complete transfer of the functional group, not only by the phosphate. So here the magnesium ion is responsible for the association and dissociation of ATP to ADP and ADP to ATP. So magnesium ion plays an important role in the energy metabolism or the activities of the high energy compound, that is the ATP. ATP is frequently described as the currency rich compound or energy rich compound. Has a high energy bond, which we studied here, and the bond nature. You analyze here some uh, primary differences here. Then what is the difference? Because at the ATP level, one ATP when released, that is the in hydrolyzed form, a high energy bond is one that liberated amount of energy, and when ATP is hydrolyzed, then the ATP first organization is AMP, then it associated to form ADP, then it associated to form ATP. And the one ATP, if hydrolyzed to form ADP, and in this process it releases 73 hundred calorie or 30,500 joule energy as the free energy. It is released in the system, that is in the form of the free energy. But and here, if it is hydrolyzed, ATP can be hydrolyzed exothermically to produce adenosine monophosphate. That means the two high energy functional groups, ADP, one phosphate group of the ADP and one phosphate group of the ATP, are released. So ATP can be hydrolyzed exothermically to produce. Exothermically means it is responsible for the release of energy, and exothermically to produce adenosine monophosphate and pyrophosphate, that means the inorganic phosphate group, and release 8,000 calorie moles, that means the 8 kilocalorie per mole, which I was discussed here. That means the ATP, I was also described in the probably in the class, as I remember, that ATP is released 7.3 kilocalorie and ADP released 8 kilocalorie. ATP to the conversion of the ATP to ADP is responsible for the release of 7.3 kilocalorie and the conversion of ADP to AMP is responsible for the release of 8 kilocalorie. And here, likely I am saying that the hydrolysis of ATP to ADP is responsible for the release of 7,300 calorie, and the ADP to AMP is responsible for the release of 8,000 kilocalorie per mole in the system. That is the exergonic reactions on it is the energy is released in the system. Thus, by exothermically, hydrolyzing can be used to power endothermic processes. It is the basic difference. So I am just, when I am coming on the next topic, some differences is here, and due to such differences, some books write or we observe the variation of the energy in the some books. And that means here you again remember that when exothermically is ATP is hydrolyzed, it is responsible for the release of free energy 7,300 calorie or 7.3 kilocalorie when it converts from ATP to ADP and releases 8,000 kilocalorie or 8 kilocalorie energy as a free energy when it converts from ADP to AMP, and the energy is used to power endothermic processes. When we see next step that the hydrolysis of ADP to AMP is also exothermic but does not occur frequently. It does not occur frequently. It may be the exothermic, but it cannot occur frequently. This indicates that uh, that is the metabolic reactions, and it indicates that the group attached to the bond when transferred to an appropriate acceptor can transfer larger quantity of energy. Here it is not release the energy, only phosphate is not, but here it is clear that as a group, it is not phosphate, but as a group, phosphate group is transferred and it is responsible for the release of more quantity of energy. So here we use the term group transfer. We use the term group transfer. Not it is responsible for the release of single bond, but it is responsible for the release of complete group. So it is referred as the group transfer potential. It represents the group transfer potential to indicate a high energy bond. Now, cells also contain other high energy compounds in addition with ATP. ATP is the high energy compound, but in addition with the ATP, some other molecules, biochemical molecules, are also present, responsible for or considered as the high energy compound because it is responsible for the release of more than 21 kilojoule per mole energy in the system after hydrolysis. So we considered these other biochemical molecules as also the high energy compound. Some high energy compounds are phosphocreatinine, and phosphocreatinine is responsible for the release of energy 43.1 kilojoule per mole. That is more than the 21 kilojoule in the system. Next high energy compound is the phosphoenol pyruvate, which we all studied during the oxidative breakdown of the glucose. That phosphoenol pyruvate is the another high energy compound, and phosphoenol pyruvate is responsible for the release of 61.9 kilojoule energy per mole. Next one is the acetyl coenzyme A. The acetyl coenzyme A is responsible for the release of 33.6 kilojoule per mole. And next one is the acetyl phosphate. It is responsible for the release of 43.9 kilojoule per mole. So some others biochemical high energy compounds are also present, which we are discussing just sometime after some time. So that means not only the ATP, some other biochemical molecules are also present in the living system, and they are considered as the high energy molecules, high energy compounds, as they are responsible for the release of free energy more than 21 kilojoule per mole. And among these four, which we are representing just, that is, one is the important, that is the phosphocreatinine. And the phosphocreatinine is an important phosphate storage compound, particularly utilized in skeletal muscles. And because the skeletal muscles are responsible for the flexibility of the body, so the phosphocreatinine is the high energy compound which is responsible for the release of a free energy during the muscular movement or during the movement of the skeletal muscles. And the phosphate group is added in this condition. The phosphate group in the condition in this condition means the in the phosphocreatinine, the phosphate group is added directly and reversibly to creatinine, directly and reversibly to the creatinine from ATP under the enzyme creatinine kinase. So, during the muscle work, ATP is needed for the muscle contraction, and at rest, ATP is concentrated in the high and the forward reaction is occurred. During the movement, the creatinine, phosphocreatinine breaks. It provides instantly energy to the muscles, and when at rest, the ATP is accumulated, and that is responsible for the directly association with the creatinine, and now it is responsible for the formation of the creatinine, phosphocreatinine. So here the association and dissociation of the creatinine and phosphate from the ATP is direct and reversible in the presence of the creatinine kinase enzyme. The hydrolysis of one molecule of phosphocreatinine liberates 3,100 kilojoule of energy. So it has the high energy bond, that is the 43.1 kilojoule, not 43.1 kilojoule, and the 43,100 joule. Here the phosphate group is associated with nitrogen, creatinine, nitrogen of the creatinine, and the phosphate group is associated with the nitrogen of creatinine to form a amide linkage. So here the energy present not present in the form of the phosphoester bond or the ester bond, but here the energy high energy bond is the amide bond. So it is the basic difference between the phosphocreatinine and ATP, because the organization and molecular organization and the nature of the high energy bond both are different. So when uh, we go through the different high energy compounds, so what is the important? That is the free energy. The free energy present in the system, either in the form of the kinetic energy or in the form of potential energy. That means in the living system, the potential energy is present in the form of bond, which I said earlier. So here the uh, if we go through the glycolysis, and the midpoint is the energetics of the ATP, the value which is responsible for the, because the in ATP is the, it is important to clear a concept here, that ATP is a midpoint. It is responsible for the accepting of the free energy, and it is also responsible for the release of the free energy in the living system. So it is considered as a baseline. The free energy present in the ATP is considered as a baseline. And the free energy of ATP, that is delta G naught dash. G naught dash means at pH normal, that is the pH value 7. And the ATP delta change in the extender free energy at normal temperature, pressure and pH is 8.8 kilocalorie. And when the components which has more than the this value, that is the minus 8.8 kilocalorie, is considered as the high energy compounds, and the components in the metabolic reactions, for the metabolic reactions. And when the energy or the value of the uh, delta G naught dash is less than the ATP, that means the less than the 8.8, it is considered as the low energetic molecules. So when the metabolic activities is occurred in the low energetics value, any low energetics value, then ATP provide energy. And when the metabolic activity is associated with the molecules having high value of the change of the free energy, standard change of the free energy from the ATP, it is responsible for the release of energy and synthesis of the ATP. So the ATP plays a key role in the living system, either for the receiving of energy or for the release of energy, which I just saying that in ATP is mediated both exergonic and endergonic reactions. In both reactions, the ATP plays a significant role. For example, I am giving some example for the proper understanding of, for the your easy understanding. You remember that the glucose 1,6 phosphate in the glycolysis, the fructose 6 phosphate in the glycolysis, the glucose 6 phosphate in the phosphorylasis, the glycerol 3 phosphate in the glycolysis. All these molecules have in D G naught dash has less than the ATP. So when the enzymatic reactions and metabolic reactions are associated with all these compounds, because they have the less free energy value than the ATP, then they require the ATP for the further activities. That means it is the endergonic. And when we remember that 3-phosphoglycerate, carbonyl phosphate, phosphoenol pyruvate, then they have the high energy D free energy value at the normal temperature, normal pressure, and normal pH. So here we studied that the ATP are synthesized from the ADP. That means in these reactions the nature is exergonic. So the ATP plays a very significant role in the maintenance of the both endergonic and exergonic reactions, and it plays a role of energy currency in the biological system. So the ATP is one of the most important high energy compound, plays significant role in the all living systems. Now, in order to maintain living processes, all organisms must obtain supplies of free energy from the environment, like autotroph, just I am saying, autotroph is directly coupled, and release of the energy, and both supply and release are coupled. I explained in my last class the coupled reaction or coupling reactions. And in this process, considered in this process that one enzyme is involved, that enzyme is adenyl cyclase. And adenyl cyclase, the enzyme not adenyl cyclase, sorry, very sorry, adenyl kinase. Adenyl cyclase is associated with the hormone mechanism. Sorry for that. Adenyl kinase. Enzyme adenyl kinase is present in most of the cells. And it catalyzes the interconversion of ATP and AMP on one hand and ADP on the other. Here some interesting facts are present. Adenyl kinase, the activity of adenyl kinase is related with the interconversion of ATP and AMP and ADP. Here, 1 unit of ATP in association with 1 unit of AMP. That means 1 unit of ATP and 1 unit of AMP. If both are associated in the presence of adenyl kinase, then 3 phosphate group and 1 phosphate group, 4 phosphate group, and it forms 2 units of ADP. So at first, the ATP, 1 unit of ATP and 1 unit of AMP are combined to form 2 units of ADP in the presence of enzyme adenyl kinase. And then the reaction which we are observing here has 3 phases. The first, it allows a high energy phosphate because AMP has low energy phosphate, that is 3 kilocalorie. It has a low energy uh, bond, and when it converts into the ADP, then it has one high energy bond. In ADP, one high energy bond. So the AMP, the low energy bond of AMP is converted into high energy bond by converting 1 unit of ATP into 2 unit of ADP. So the reaction allows high energy phosphate in ADP to be used in the synthesis of ATP. The ADP and ADP, that means it has 2 unit of high energy bond, then 1 unit of high energy bond is transferred to the ADP, and it is responsible for the synthesis of ATP. It is the first significance of this reaction. The second significance, it allows AMP to be recovered by rephosphorylation of ADP. Suppose AMP is decreased in the body, so 2 unit of ADP is rephosphorylated to form 1 unit of ATP and 1 unit of AMP. So it is second significance. And third significance is it allows AMP to increase in contraction. To increase in concentration. To increase in concentration. When ATP becomes depleted, because after the synthesis of the ATP utilized in the metabolic activities, and it converts into the ADP. So when it is depleted, the ATP is depleted, then the AMP acts as the metabolic or allosteric signal. I will again discuss the allosteric process when we will discuss on the enzymology or kinetics, enzymatic kinetics. And here the AMP AMP is acts as the allosteric signal to increase the catabolic reactions. And when the catabolic reactions is increased, then it is responsible for the production of more and more ATP. And then the ATP is responsible for the functioning as the energy currency. So the depletion of the ATP increases the concentration of AMP, and then AMP acts as the allosteric signal, which is responsible for the initiation of the catabolic reaction and further production of the ATP. So here these three significant functions are associated with the energy ATP, action of the ATP, action of the enzyme on the ATP. Now, as we suppose that you are understanding properly all these facts. Second one, I told you that the not only ATP, some other energy rich compounds are also present in the living system. I am just describing all these concepts in the present live class, but two more important high energy compounds are I am discussing here. That first is the guanosine triphosphate and GTP. The guanosine triphosphate or thymidine triphosphate or cytidine triphosphate, of course, the thymidine triphosphate and guanidine triphosphates energetically they are synthesized but have no more biological significance, so I am not describing here. But of course, all these four nucleotidic components, that is adenosine triphosphate, guanosine triphosphate, thymidyllic triphosphate, and cytodyllic triphosphate, all these properties are present, and of course, all these components are used during the molecular activities at the DNA of the DNA and RNA here. But we I am discussing on the energetics part, so we are not I am not describing here the TTP and CTP here. But the guanosine triphosphate is significant, and the guanosine triphosphate, the synthesis nature is same, and the guanosine triphosphate is associated with the cellular process and protein synthesis, associated with the cellular process in the protein synthesis and signal transduction. In both these processes, the energetics value or the energy transport of the energy transported by the GTP is significant. And I will discuss these two topics when I will study. I will study the signal transduction and protein synthesis. The next one is the high energy compound. The next high energy compounds are NADH, that is nicotinamide adenosine dinucleotide, and FADH, flavine adenosine dinucleotide. These two compounds are also high energy compounds, and the high energy compounds, the nicotinamide adenosine dinucleotide, that is NADH, and flavine adenosine dinucleotide, that is FADH. And as I suppose that you all know that both these NADH and FADH are associated with the electron transport system, and you all must know these two components. And here the high energy, these two high energy compounds are associated as the electron carrier. And when if you remember something, that when the electron energy, if you observe the release of energy at the molecular level or at the atomic level, not at the molecular level, but at the atomic level, then it is very clear that energy is released when energy electron is transferred from higher energetic level to the lower energetic level. And you all know it very well from the quantum mechanics. And when the energy passes from lower energetic level to the higher energetic level, it requires some energy from the source, other source. So here, the when the electron transport system is, we study the electron transport system, we easily observe that the NADH and FADH are responsible for the carrier of the H plus in the form of, and the H plus is released, and the release of the H plus is associated with the energetic sense of electron during the electron transport chain. So these two are significantly associated with the electron transport chain in living system. Next, which why I want to introduce, that one bond is the phosphoester bond, and not the phosphoester bond, but phosphate as a group is considered as the it is more better to say that not the phosphoester bond, but the phosphate as a group is responsible for the high energy level. And likely, that, like in the phosphocreatinine, we observed that the phosphate group is associated with the nitrogen, not with the carbon. The phosphate group is associated with the nitrogen, and that group is responsible for the nature, acquires the high energy nature. Likely, in the biological system, which we observe, and just I am saying a different number of names like coenzyme A, glycerol 3 phosphate, phosphoenol pyruvate, all these biochemical components have high energy. That bond, that means the group. Bond means not a single bond, but as a group. So the high energy bonds we commonly refer to the high energy bond, and the high energy bond is, if you consider in the ATP, it is referred as the phosphoanhydride bond. It is the phosphoanhydride bond. In the phosphoenol pyruvate, that is enol phosphate bond. In the acetyl coenzyme acetyl phosphate bond, we studied in the electron transport system that simply the acetyl group of SH that is associated, that is the thiol bond, and it is associated with the synthesis of the acetyl coenzyme A. But it is not a simple process. It energetically it is completed in the at the five stages, in the five stages. So we are not discussing here in detail, but we just want here, I just want to know the what is the high energy bonds, and you remember all these things further. The enol phosphate bond, that is the phosphoenol pyruvate. In the phosphoenol pyruvate, that is the enol phosphate bond is present. In acetyl coenzyme A, acyl phosphate bond is present. In phosphocreatinidine, guanidino phosphate bond is present. In acetyl coenzyme A, thioester bond, that is the SA, is present. And likely in the biological system, these are considered as the high energy bond. So, as I suppose that you all know the what is the high energy compound, what is the high energy bond, what is the energetics of the high energy bond, and what is the significance of the free energy in the synthesis of the high energy bond, how ATP is associated with both endergonic and exergonic process. Now, I give thanks to all of you who are associated here. And if you have any question, you are free to ask and give me message. I am waiting here for 2 or 3 minutes. If you have any question, you ask or give me message. Probably no one asked any question, and thank you to all of you. And tomorrow I give you message. I will give you message for the class, that the time of the class is same, that is 8 p.m. But I will communicate you the topic of tomorrow. Thank you. Thank you to all.